So let's get started the seminar for today. So it is my great pleasure to introduce Dr. Largaespada, Dr. David Largaespada. He's been a long-time member of Masonic Cancer Center, and he's a graduate from University of Minnesota and did his PhD at Wisconsin. And after doing postdoc at the NIH, he became a faculty of this university. And since then, he has assumed many positions. And he's currently the full Department of Pediatrics and GCD full professor and co-director of brain tumor program and the Center for Genome Engineering. And also he holds a Harvard Family Children's Cancer Research Fund Endo Chair and McKnight Professor. So then actually, everybody knows him, on campus. So then I don't have to give a long introduction, but one thing which impressed me is when I have an online, the meeting with him, the background of him has a lot of empty champagne bottles. And that is a kind of every time celebrate the graduation of PhD, he opens the bottle. It means that it's a kind of trophy for him. They're also, they're not only good, being a good researcher, and he's a really great educator. And many current council members are educated by him for PhD. And so that I really appreciate his contribution to Masonic Cancer Center. And I'm looking forward to hear your talk today. Thank you.
Thanks, Masato. Well, thanks, everyone. I'll jump right in. So these are my disclosures. So I have co-founded a few biotech companies and worked with some others, but none of the presentation today is really relevant to that work. So, my lab is basically a cancer functional genomics lab. That's what we do, with a focus on nervous system tumors and sarcomas. And overlapping this is tumors that are common in a cancer predisposition syndrome called NF1, or neurofibromatosis type 1 syndrome, which is a major focus of our lab. As far as the methods that we use, we've spent a lot of time trying to build better models to do functional genomics and test new therapies. So we do cell line screens using CRISPR and small molecule screens, in vivo screens using transposon mutagenesis. And I'll talk a little bit about all three of these things. And then finally, using these model systems, testing new therapies that are suggested by the functional genomic screens that we do. So today I'm going to talk about small molecule and CRISPR screens that we hope have allowed us to figure out how we can leverage synthetic drug sensitivities that occur as a consequence of loss of certain tumor suppressor genes in sarcoma. And if there's time, I also want to talk about something new in the lab I'm excited about, which is transposon screening for improved adoptive cell therapy. So in particular, how we're doing transposon screening to try to identify pathways that we could alter to prevent tumor-associated T-cell exhaustion. So this all starts by thinking about the fact that identifying cancer drivers has had profound implications for therapy. Everybody knows the story of Herceptin, developed to treat a subset of breast cancer cases that have amplification of HER2 oncogene. And this has led to the concept of precision medicine, where if you understand the genetic alterations present in a tumor, you can predict what therapies might be useful. And it's in fact an FDA-approved concept. And there are companies like Foundation Medicine that will genomically screen your tumor, looking for actionable alterations that could tell the oncologist what therapy to prescribe. The problem is that many cancers haven't benefited from this precision medicine idea. So, and this includes sarcomas and many childhood solid tumors. And, I think this fact is exemplified by the story of Zachary Bartz. He's a patient we knew who had neurofibromatosis type 1 syndrome. He and his family became champions for NF1 research in Minnesota. He suffered from optic pathway glioma, which is a benign brain tumor that he was treated for by Chris Mortel here at the university. But unfortunately, he developed an aggressive sarcoma and passed away at age 11. And his case exemplifies the problem this type of sarcoma and other childhood sarcomas can't easily be treated once they become disseminated throughout the body. They don't respond to currently available immunotherapies. And also, genomic analyses of these types of sarcomas doesn't reveal clues about how to treat them. So they're genomically unstable, have P53 alterations, but they're not characterized by activated oncogenes that you could target using precision medicine. And in fact, most pediatric tumors lack obvious target for therapy based on omics data. So this is from a 2018 consortium paper published in Nature. A few types of pediatric solid tumors do have high frequency of druggable target alterations, but few of them have really been validated as druggable. Most are potentially druggable alterations. And so even clinical trials that have been done in pediatric solid tumors where there's an alteration in the RAS/MAP kinase pathway, and you might expect would respond to MEK inhibition, so far they've all failed. So we have a problem. How do we find ways to treat cancer that has few or no truly actionable mutations? And so my lab is interested in this problem. It's a problem not just for pediatric cancer, but probably some of you know friends or family who've developed cancer, adults, and they got a report from Foundation Medicine, and it doesn't really say anything particularly useful. So that's the situation we're in oncology right now for many patients. So one thing that we're doing is trying to pursue this idea that there are useful drug targets you could find if you could understand copy number or epigenetically altered tumor suppressors and oncogenes. You know, this is the kind of information you don't typically get with, present day omics screens. And for this, we've been doing transposon mutagenesis in mice to try to understand pathways that lead to cancer by using the mouse and the mouse genome as a model for human cancer. And I won't really talk about that today. A second idea we're pursuing is that many of these tumors are characterized by recurrent loss of certain tumor suppressor genes, and that might provide avenues for therapy. The other thing that we've started to do is to look at immunotherapy for these types of cancers. And we're pursuing vaccines based on neoantigens from misspliced mRNAs, oncolytic viruses, and adoptive cell therapies. And I'll talk a little bit about these things today. One other way to think about what we're doing, and this is a figure that Julia Nickrad in the lab made, so I got to thank her for it, is try to kill tumors from the outside and the inside. What are the intrinsic vulnerabilities that they have? And also, can we exploit extrinsic vulnerabilities, namely trigger a therapeutically useful immune response? Those are the things that we're working for. Trying to trigger this effective immune response or create an effective immune response with adoptive cell therapy. And for the intrinsic vulnerabilities, finding small molecule combinations that could selectively kill mutant cancer cells. So I mentioned already that a big focus of the lab is this cancer predisposition syndrome, NF1. It's an autosomal dominant syndrome affecting about 1 in 3,000 individuals, so it's fairly common. 50% of all cases are de novo, meaning the affected patient is the first in their family to have it. So this disease can really affect any family. All patients who have NF1 syndrome have cafe au lait spots, Lish nodules, which are hamartomas on the iris, and dermal neurofibromas, and this helps in diagnosing the disease. But it's a complex syndrome, and their patients can be affected by many other things: skeletal, endocrine abnormalities, problems with learning and memory, ADHD. From the standpoint of cancer, the disease is important because they're prone to benign and malignant tumors. And in fact, the most common cause of disease-associated mortality is the development of malignant peripheral nerve sheath tumor, or MPNST, an aggressive soft tissue sarcoma. Now, the disease is caused by inheriting a loss of function mutation in the neurofibromin, or NF1 gene. The protein product of the NF1 gene is a RAS GTPase activating protein. It's a negative regulator of RAS signaling. So you can imagine when it's lost, RAS is going to accumulate, active RAS will accumulate in the cell. And this is an important tumor suppressor gene outside of NF1 syndrome, since you see alterations in NF1 that occur in sporadic cancer also. And so understanding this disease is important for understanding sporadic cancer as well. Now, NF1 associated peripheral nerve sheath tumors develop from the Schwann cell lineage. Schwann cells are these glial cells in the peripheral nervous system that myelinate axons in the peripheral nervous system. And it's sort of thought that the peripheral nerve sheath tumors associated with NF1 syndrome, the target for these tumors may be some Schwann cell precursor. It's kind of unclear exactly when in the development of the Schwann cell these alterations occur. We do know that MPNSTs develop along a really specific histopathological pathway. So as I mentioned, the patients are heterozygous for a loss function mutation in NF1. That's why they have the disease. And in a rare Schwann cell or Schwann cell precursor, there's loss of the wild type copy of NF1. And that, along with other changes, can lead to a plexiform neurofibroma, a deep nerve form of the neurofibroma, different than the cutaneous ones, and that are a real problem. Then there's, within that tumor, there can be a cell that loses CDKN2A or CDK2A and 2B, and you get an atypical plexiform neurofibroma that's considered pre-malignant. And then that can progress to MPNST, and that's usually associated with loss, biallelic inactivation, or loss of EED or SUS12, which are critical components of the polychrome repressor complex 2, which I'll talk about a little bit more about later. And so it's this stepwise process. And so you can think of MPNST as developing by the stepwise loss of tumor suppressor genes that are removing barriers to malignancy. And so this is sort of a challenge to treat then if they don't typically have activated oncogene like BRAF that you could target. It's instead the loss of these tumor suppressor genes. Well, what about current treatment? So surgery is used oftentimes. The tumor's in a place where you can't get clean margins. Radiation and adriamycin-based chemo, but local recurrence is common, eventual development of metastatic disease, and half the patients are dead by 4 years. And this would be a typical Kaplan-Meier for overall survival and development of metastatic disease. There's been many failed recent clinical trials for MPNST. So we're really looking for something new in the field. Most of these clinical trials have tested signal pathway inhibition, but none of them have worked. I will say though, given what we know about the function of neurofibroma and the NF1 gene, RAS activated signaling pathways are likely to be useful targets for therapy. So remember that NF1 or neurofibromin is a RAS gap. So when it interacts with RAS GTP, it increases RAS's intrinsic GTPase activity 1,000-fold. That GTP is converted to GDP, and because RAS cleaves off that third phosphate, and it becomes inactive. So if neurofibromin is gone from a cell, RAS GTP levels rise, and you get increases in RAS activated pathways like MAP kinase pathway, PI3 kinase pathway. And so it makes sense that targeting these pathways might be part of treating NF1 associated tumors. And one molecule downstream of RAS GTP that's attracted a lot of attention is MEK, downstream of RAF after RAS activation. And in fact, a MEK inhibitor, selumetinib, is active for plexiform neurofibromas. This is data taken from a paper published in New England Journal of Medicine showing that the MEK inhibitor selumetinib taken every day could reduce the size of plexiform neurofibromas in patients that carry that benign form of the Schwann cell tumor. And that led to FDA approval of selumetinib for plexiform neurofibromas in 2020. More recently, Chris Mortel here at the University of Minnesota and SpringWorks, a pharmaceutical company, has showed another MEK inhibitor, mirdametinib, might work even better, where they get tumor shrinkage in most patients. And all of this has been great. And this is, the first real medical therapy that we had for NF1 syndrome. But there's some other questions. Could we improve the response to the MEK inhibitor? So it shrinks the tumors, but they don't completely go away. Some patients can't tolerate the MEK inhibitor therapy because of the side effects. So could we use a lower dose and get a better response? Would they be useful for treating MPNSTs? So far, the answer is no. By themselves, they don't. They don't work for the malignant form of the disease. What other pathways are likely to be usefully targeted in MPNSTs? What other vulnerabilities do NF1-related tumors have? And so those are the things I want to talk about first. And I want to talk about this idea of synthetic lethality. And we've been pursuing this in NF1-associated peripheral nerve sheath tumors using isogenic human cell lines we've made where we've either knocked out NF1 or not, as well as other tumor suppressor genes. And so this part of the talk really is on this idea of targeting intrinsic vulnerabilities or cell autonomous in NF1 syndrome-associated tumors. So what is synthetic lethality? This is an incompatibility of cell survival with the loss of function of two or more genes in a single cell, where defects in the single genes involved in this interaction do not lead to cell death. So imagine that you have a patient with NF1 syndrome. They're heterozygous for the NF1 mutation, but they develop a tumor. It could be the benign tumor, or it could be the malignant tumor, but both copies of NF1 are gone now. We'd like to know what genes have a synthetic lethal interaction with NF1, where you get a non-viable cell if both copies of that gene were also mutated. Whereas in the wild type cell or the NF1 heterozygous cell, loss of both copies of that gene would not kill the cell. So that's what we're looking for. And we know the function of neurofibromatosis includes the fact that it has this gap-related domain in the middle, but it has functions other than acting as a RAS gap. And we cannot predict easily what sensitivities may result from loss of neurofibromatosis expression, and the synthetic lethal effects are likely to be context dependent. So our thought when we started this work was, let's take human Schwann cells and knock out NF1 or don't, and then see what drugs these cells become sensitive to and what genes they become dependent on. And so this is work from Kyle Williams, a postdoc in the lab that he's led along with the help from many others. And he's taken human immortalized Schwann cells and left them wild type or made sister clones where he's knocked out NF1, both copies of NF1, and done a drug and small molecule screen to look for drugs that selectively kill NF1 deficient cells and not the wild type isogenic control cells. So he used CRISPR to do this and introduced indel mutations in exon 10. He was able to isolate multiple clones that no longer express neurofibromin protein by Western blot and have biallelic insertion mutations. These are sister clones that are wild type and retain expression of NF1, but which went through the same process, CRISPR screening. The NF1 knockout cells have a higher basal level of RAS GTP, as you would expect based on what we know about the function of neurofibromin. These cells are also poised for transformation in the sense that NF1 knockout cells can form colonies in soft agar under low serum concentrations, more so than the wild type controls. And some cells within the NF1 deficient population are capable of forming tumors. When you inject them in nude mice, a subset of the cells will grow into a tumor. So we felt confident that we had a good model system that we could use for doing this drug and small molecule screen. And we worked with Gunda Georg and John Hawkinson in the Therapeutics Discovery and Development Institute here at the university to screen about 11,000 drugs, looking for ones first that had activity versus NF1 deficient Schwann cells that reduced down to about 600 or 700 drugs. Those 600 or 700 drugs, we then did an 8-point dilution screen and looked for the IC50 in wild type human Schwann cells and isogenic NF1 deficient human Schwann cells, looking for a shift in the IC50. And by doing this, he came up with drugs that kill NF1 mutant Schwann cells more readily than wild type cells. This is a list of the drugs that he found. And they fell into certain activity classes, making us think that this whole screen was working. So for example, there were drugs that increase intracellular calcium or affected the NF-kappa B pathway. So then Kyle took representative drugs from each of these classes and tested them further in vitro and then in vivo. And these drugs showed promise both in a genetically engineered mouse model of the plexiform neurofibroma, as well as in xenografts of human MPNST cells grown in immunodeficient mice. So this particular mouse model of a plexiform neurofibroma was developed by our lab and published a long time ago. But basically what we have is deletion of NF1 in Schwann cell lineage cells and P10. And these mice developed, when we did this, these mice developed 100% penetrant multifocal plexiform-like neurofibromas, and so many that it actually caused lethality in the mice around the age of weaning. Now, our justification for doing this was that P10 is downregulated in human plexiform neurofibromas. And so this allows us to have a mouse model that develops these tumors really fast. So we actually treat the mice with drugs before weaning, and we can get an answer about whether the drug's working in a genetically engineered mouse model really quickly. These are standard xenografts. So just as an example of what we saw, one of the drugs that we found was a cardiac glycoside called digoxin, which is used to treat heart failure, and that was effective in the MPNST xenograft model. Here you can see tumor volume versus days after enrollment in the study, and mice got no treatment or vehicle alone or digoxin, and you can see this works as we predicted it might. It can reduce tumor size in some of the mice and prolongs their survival. And Kyle must have got lucky because every drug he picked worked in both the genetically engineered mouse model and the MPNST xenografts to some extent. So this is the genetically engineered model of plexiform-like neurofibromas. This is untreated. As I said, these mice develop aggressive multifocal plexiform-like neurofibromas and die around weaning. All of the drugs he picked from the different classes extend survival to some extent, and the same was true of MPNST xenografts. Obviously, though, this isn't good enough. So we thought about, since our, a big target for us was MPNSTs, can we also exploit the loss of other tumor suppressor genes that happen during the development of MPNST? I mentioned that most MPNSTs lose the polychrome repressor complex 2, and that's usually by biallelic inactivation of SUS12 or EED, critical components of this complex. This complex is important in maintaining cellular identity. It mediates the mono, di, and trimethylation of histone H3 at amino acid position 27, lysine. So MPNSTs and some other forms of cancer will also show loss of the PRC2, usually by loss of SUS12. This histone H3 lysine trimethylation mark is important for the repression of many genes. So when it's lost from MPNSTs, you get de-repression of gene expression of hundreds of genes, which we've studied, but I won't talk about today, that are involved in a lot of different pathways, the Wnt pathway, sonic hedgehog, notch signaling. And so we thought, well, perhaps loss of the PRC2 also exposes vulnerabilities that can be leveraged to treat these tumors. And so Kyle made both NF1 wild type and NF1 deficient cells further deficient in SUS12. So this Western blot just shows that he got multiple clones with loss of SUS12. They show loss of histone H3K27 trimethylation. Interestingly, they show a trend toward a gain in acetylation of that same residue. So histone H3K27 can be methylated, and that's associated with gene repression, or acetylated, and that's associated with gene expression. And so he then did a screen of SUS12 deficient human Schwann cells on an NF1 deficient background versus NF1 deficient alone. And he used a library of epigenetic drugs. Since the PRC2 is an epigenetic writer, we sort of thought that drugs that target the epigenome might be useful. And what he found out was, and this was about 240 drugs or so, that certain drugs scored. Histone deacetylase inhibitors, aurora kinase inhibitors, some DNA methyltransferase inhibitors. And this just shows an example where an HDAC inhibitor called Verinostat, you see percent cell viability versus concentration. And you can see controls up here that retain SUS12 or isogenic SUS12 deficient. And at a certain concentration of Verinostat, the cells just start dying. So this effect isn't subtle. It's quite pronounced. And we see it whether we use a knockout that we made by CRISPR. We see it if we instead use siRNA to knock down SUS12 or EED. We see it if we use an enzymatic inhibitor of PRC2, which is an EZH2 inhibitor. We've seen it. Other people have seen it too. And so I really believe this, and it's robust to multiple cell types as well. Histone deacetylase inhibitors are well known in the cancer field. Several pan-HDAC inhibitors are FDA approved for T-cell lymphoma and multiple myeloma. They've been tested in many other cancers. It's unclear how they exert their anti-cancer effects. But because the histone deacetylases are involved in transcriptional repression, since they remove the acetyl mark associated with gene expression, by inhibiting the HDAC, you might cause increased transcription of genes. And if those are tumor suppressor genes that you're reactivating in these tumors, that might be how they work. In NF1 SUS12 deficient cells that we've made, when we add the HDAC, what we see is increased chromatin-associated HDAC2, increased H3K27 acetylation, increased P27 and markers of apoptosis. And so when SUS12 is gone, the cells have higher basal levels of histone H3K27 acetyl, which makes sense because that residue can't be methylated. So instead it's acetylated. Now, when you add the HDAC inhibitor, Verinostat, the levels of histone H3K27 acetylation go even higher to some toxic level sooner than wild type cells do. So that's our explanation for why PRC2 deficient cells have this hypersensitivity to HDAC inhibitors. This very high level of histone H3K27 acetylation probably messes up transcription of certain genes. And we see, for example, decreases in MYC and other genes. So it's sort of paradoxical that you're doing something that exacerbates the effect of PRC2 loss, but it brings it up to some toxic level. And this is sometimes referred to in the field as transcriptional toxicity. The HDAC inhibitor Verinostat is highly active in MPNST xenografts. This shows what Verinostat can do in terms of compared to no treatment in an MPNST cell line xenograft. It dramatically extends survival. So then we started doing very simple-minded experiments. Since we have two drugs that might work, MEK and HDAC, let's just combine them. When you combine them in vitro, they do show formal synergy as shown by determining a combination index. And they show synergy in terms of the xenografts. So this is the cell line xenograft data. You can see here an example of that where these are the single, this is no treatment, the single agents or combining them. And it works with cell line xenografts, induces massive apoptosis. It can actually cause a reduction in tumor size in vivo. And the combination also works versus patient-derived xenografts. And we've done this in multiple patient-derived xenografts where the combination works much better than the single agents. And so this has led to trying MEK inhibitor plus HDAC inhibitor off-label here at the University of Minnesota by Chris Mortel, who sees MPNST patients. So this is the experience with one particular MPNST patient that he's treated with a combination of these drugs. This patient had unresectable brachial plexus H3K27 trimethyl negative MPNST. So this shows that the patient had PRC2 loss in the tumor. This patient had failed radiation therapy and doxorubicin-based chemotherapy already. At that point, there's no standard of care for an MPNST patient. And so Chris felt it was okay to use these two FDA-approved drugs off-label for this patient. And so he came up with a way to combine the two drugs together. The patient has resolution of pain associated with the tumor in the shoulder, significant reduction in PET SUV max, which is a measure of the metabolic activity of the tumor. The combination was well tolerated. And because of this, we're now working on a IND to the FDA for a window of opportunity trial where we would treat patients like this for a month, do biopsies before and after to see if the drug's hitting its target and see if the tumor's responding by doing that. This patient's still alive and doing well and on therapy. So, you know, that's promising, but, you know, we might need other drugs. And so Julia Nickrad and Ethan Novosek in the lab have also taken the same cells I just talked about and done whole genome CRISPR screen to look for synthetic genetic lethality associated with gene mutations. So to do a CRISPR screen like this, you take wild type cells and isogenic knockout cells and transduce them with a lentiviral library. And you've engineered these cells to express Cas9. You transduce them with a lentiviral library with guide RNAs versus all genes. It has a puromycin marker, select in puromycin, grow the cells for a certain number of cell passages, usually at least 8 doublings, population doublings, isolate the cells, PCR amplify and sequence all the guide RNAs that are present in the wild type cells and in the mutant population and compare that data to look for candidate genes that must have caused synthetic lethality in the mutant population because those guides are missing from this population, but present in the wild type cells. So that's what we're looking for. So there's a lot of work that has to be done to do one of these. Sorry. Synthetic lethal screens. PowerPoint's not responding. I think I clicked it too many times or something. Yeah, it doesn't want to move to the next slide.
Hmm.
All right, thank you. So just, I'm not going to go through all this data, but if you want to do one of these screens, come and talk to us, because we made a lot of mistakes doing these the first few times, but you have to make a cell line that has a uniform high-level expression of Cas9 when you do this first, and thoroughly validate it by flow cytometry using a vector that expresses a Cas9 T2A BFP or GFP fusion. You have to do Western blots and you have to test it to make sure that when you add a guide RNA, you get good cutting at a test site. But you can do this whole thing and isolate the DNA and from the cells and go through this process called MAGIC, which allows you to find guide RNAs that seem to be depleted selectively in your mutant cells compared to wild type ones. And so when Julia and Ethan did this, they found shared and genotype specific negatively selected guide RNAs for genes. So they did this in wild type Schwann cells, isogenic derivatives that had loss of NF1 or loss of NF1 and SUS12. And this shows the number of guides that were unique to each genotype or shared by genotypes. So one way of looking at the data is like this, where you look for a fold change in guide abundance for genes in wild type versus NF1 deficient, for example, or wild type versus double deficient cells. And the majority of genes, their guides are about at 0-fold change in this big cloud for both populations. Some guides are depleted in both populations, and some guides will be selectively depleted, for example, here in the NF1 deficient cells, but not wild type cells. And so that's how you can get gene lists that you can work with from this type of study. And rather than go through a lot of individual genes, I'll just show you that they did fall into certain pathways. So synthetic lethal genes associated with RNA splicing and processing were found in NF1 null cells, DNA replication, chromosomal organization, and cell cycle in NF1 SUS12 deficient cells. And so to follow up on these studies, Julia came up with a medium throughput synthetic lethal candidate gene pipeline where she uses the same Cas9 expressing cells she has before and puts in a single lentivirus with a single guide in it, does the same puromycin drug selection, and then just stains the cells at a later time point to see if any cells survived. And as a control, we can use a scrambled guide, a guide against a non-essential gene like HPRT1, or a guide at AAVS1. And as a lethal control, we use guides versus PCNA, which every cell needs to survive. And so you can see when you add a guide with PCNA, it kills all the cells. And these results are in wild type cells. And you don't see much of an effect on these candidate genes, IMP4 or NDUFV1. Whereas at least IMP4 scores as selectively as a synthetic lethal candidate for NF1 deficient cells. And so we can get data like this where we look at a bunch of candidates and what we found is that this gene, which is involved in nitric oxide production, does validate in both NF1 deficient and doubly deficient cells compared to wild type cells. This protein called KIF18A validates, which is involved in microtubule dynamics, and a drug versus this protein is already in clinical development. And so I've shown you how we can treat cancer with no actionable mutations potentially by identifying synthetic lethal genes and drugs that show a synthetic lethal effect on a mutant background. And here's some of the candidate drugs that we've identified. But I would say that going forward is really hard, in part because we have too many candidates. So we need better model systems. MPNST is a rare disease. There's no way we could test all these drugs and drug combinations in clinical trials. So we're going to have to do something. And so this is related to the idea that I brought up at the beginning, that we need better model systems. And so we've invested in making iPSC, human iPSC derived models of NF1-associated peripheral nerve sheath tumors, new genetically engineered mouse models of MPNST that incorporate loss of PRC2. Working with others, developed a bunch of MPNST patient-derived xenografts. Even a porcine model of NF1 syndrome that develops benign tumors, peripheral nerve sheath tumors. But what I want to talk about now is a method we're using to try to sort through these drugs, which we also couldn't test in vivo, all these drugs easily in drug combinations, which is 3D microtissues using cells from a PDX to do drug screening. So this is a multi-institutional collaboration between our lab, Dave Wood's lab here at the University of Minnesota, our collaborators at Washington University, Angela Irby, and at Johns Hopkins, Christine Pertillis, as well as an informaticist we collaborate with, Sarah Gosling. And the idea behind this project, which had funding from the NF Research Initiative and now from the DoD, was to create and characterize a bunch of MPNST patient-derived xenografts because they didn't really exist in the field. And then also to utilize a new technology developed in Dave Wood's lab to screen them for drugs that were of interest to the labs involved in this consortium. And I want to talk about that technology that Dave Wood's lab developed, and we collaborate with his technician, Elizabeth Rono, on this. What he found is that flow-focused microfluidics can be leveraged to make collagen-based microtissues in which you embed cells. And this is what these microtissues look like, and they're suitable for doing high-throughput drug screens. And this work was recently published in Neuro-Oncology using MPNST-derived cells, and Alex Larson in my lab spearheaded this work. He works with these technicians to expand the PDXs in mice. We create a single cell suspension. We deplete the dead cells and the mouse cells, and then put them into these microtissues. And this is an image from the paper showing what these microtissues look like. And you can stain for dead cells and living cells. And so expose them to drug, and the readout is imaging based. And you can get, when everything works right, you can get really good dose response curves that look like this. This is some data that Alex got. So you can see the percent viability with normalized percent viability versus drug concentration for Joxin, one of those drugs I talked about earlier. And so you can estimate an IC50 from this. Now we're using primary PDX derived cells, so that's better than a cell line. This isn't as good as doing it in a mouse, but it's probably better than information than we could get using 2D culture and established cell lines that have been cultured forever. And this is the same kind of data, but for doxorubicin. And what's great about this is you can get a lot of data and go through drug classes like chemotherapeutic drugs, MEK inhibitors, mTOR PI3 kinase inhibitors, RTKs inhibitors, other drugs. You can do drug combinations, and drug combination testing in the 3D microtissues in vitro can predict responses in vivo. So this is an example for one particular PDX from Johns Hopkins. And this shows normalized viability, cell viability. And we're showing the results after 2 days of drug exposure or after 5 days. And in this experiment, Alex and Elizabeth combined olaparib, PARP inhibitor, and trabectedin, a traditional chemotherapeutic drug, or a MEK inhibitor, mirdametinib, and trabectedin combinations. And after 2 days, the combo wasn't doing anything really different than the single agent trabectedin. But after 5 days, you start to see that the combination is much better than the single drug. And in this experiment, the trabectedin dose was held constant. So, but by itself doesn't do much. But in combination with the PARP inhibitor, it totally crushes these cells. Similar effect for mirdametinib plus trabectedin. And then that predicted a really good response of this tumor to the combination of these drugs as in vivo as well. So, this is a way that we hope to be able to move forward better with this work. So I'd like to know if we can rationally choose better drug combinations by considering drug interactions, metabolism, and delivery. So I don't know the answer to this, and I hope that someone here does, or someone here at the university can help us with figuring out, based on more first principles, what drugs we should be combining for this type of therapy. All right. So just in the last 5 minutes, and unfortunately I'll have to go through this fast, I just want to mention we're also hoping that tumors like these could be targeted with the immune system. So, drugs are good, but now we have proteins, viruses, and cells as drugs, spanning in size, 5 orders of magnitude. And we're working on oncolytic viruses and adoptive cell therapy as well. And these therapies are meant to leverage the immune system. The challenge is that MPNSTs are immunologically often cold. So this is an analysis of gene expression data by Robert Galvin in the lab. If you look at MPNSTs, some of them have very few T cells. Some of them have more T cells, but oftentimes they're cold. And when the T cells are there, they show markers of exhaustion. This is all based on gene expression profiling. Okay. So active infiltrating T cells might synergize with other therapies like we've been talking about today. So this raises the question of what T cell autonomous mechanisms could be modulated to improve tumor homing and tumor activity. And what I wanted to talk about, at this point was work that we're doing based on the Sleeping Beauty transposon system. Sleeping Beauty is a cut and paste DNA transposon, a 2-part tool, the transposase protein and transposon vector DNA that you can combine to get a cut and paste reaction in cells. And we use this system to study cancer development in the past, where we made mice that carry a transposon vector, cross those to mice, and it would carry multiple copies of that integrated in a single place in the genome. Cross those to mice that express the transposase, the double transgenic mice, depending on where the transposase was expressed, many somatic insertion mutations would occur in that tissue. The transposon we mobilized was created by my first graduate student, Adam Dupé, which has a strong promoter, an artificial exon with a splice donor, splice acceptors in both orientations, and a bidirectional polyadenylation site, so that it could turn genes on or turn genes off. So gain or loss of function mutations. And we mobilize that again from a multi-copy concatemer. And so using this tool, we're able to carry out screens for cancer genes, where we induce tumors by insertional mutagenesis, and we identify genes that are recurrently altered by transposon insertion in multiple independent tumors. We call those common insertion sites. And that allowed us to find known and novel oncogenes like these, as well as tumor suppressor genes. And so that's all been good, but what we're doing now is to try to use the same approach to select for cells that have non-neoplastic phenotypes, like T cells that are better at evading tumor-induced exhaustion. So to do this, we created a new transgenic mouse, and Alex Larson spearheaded this approach that expresses an improved version of the Sleeping Beauty transposase, but which allows us to express it tissue specifically and under temporal control with a doxycycline system. Okay, so to express the transposase and get transposon mobilization, you need Cre-recombinase and doxycycline plus RTTA. So Wendy Hudson in the lab bred all these mice together to create what we call quad mice that have the 4 transgenes necessary for inducible SP transposition in mature T cells. The reason for the inducibility part is if we leave Sleeping Beauty transposition going forever, the mice will get T cell leukemia. This way we can mutagenize the T cells for a while so that they have new phenotypes, but not so long that they get cancer, and select for new phenotypes. And so CJ Lee in the lab has used these T cells to screen for exhaustion and resistance. So he's taken wild type T cells or SP activated T cells, activated them in vitro, and then grown them in IL-2 alone or IL-2 plus recurrent anti-CD3, which induces exhaustion ex vivo. And when he did this, he found that the SP mutagenized T cells remain more viable under exhaustion conditions compared to wild type T cells. And that's shown here. Under IL-2 alone, both wild type and SP mutagenized T cells grow just fine. The SP mutagenized T cells have reduced PD-1 expression under exhaustion conditions compared to wild type cells, and that's shown here. And so then we did RNA sequencing on these T cells and identified genes that are recurrently altered, because he did this experiment in 2 parallel cultures. He looked for genes that are recurrently altered in exhaustion resistant cells compared to cells grown in IL-2 only. And this was bioinformatics work done by Tyler Jubinville in the lab. So these are some genes that are recurrently altered in IL-2 only or IL-2 plus CD3. These ones are unique to the CD3 exhaustion condition. So these genes would be candidate genes for mediating exhaustion resistance then. And an example is BOC2 and the insertions activating BOC2 in both cases, in both parallel cultures, create fusions with the gene upstream of the open reading frame. And so the prediction is this would result in overexpression of BOC2, the whole protein. And that's also what we see by the RNA-seq. These quad mutagenized BOC2 cells are expressing higher levels of BOC2 than the control exhausted cells. So you might notice that some of these candidate exhaustion resistance genes are leukemia genes. So should we really put them into something like a TIL or a CAR T cell? But I would argue that if you think about what cancer cells can do, and they can do all these things, we actually want adoptive cell therapies to do many of the same things, except we don't want them to proliferate without normal controls or lose differentiation, but we want them to be able to survive in circulation better, resist cell death, and evade the immune system, have extended longevity and competitive advantage versus other cells. So I think that's probably okay. And so in thinking about where we're going with this, you know, we did this screen for T cell exhaustion resistance and have candidate genes. In the future, we want to validate these exhaustion resistance genes and test them in various adoptive T cell platforms. I should mention that we use the same quad mice to do an in vivo screen for enhanced T cell homing to pancreatic tumors with Ingen Stromness's lab. And the results there are very interesting. And we have candidates that seem to be able to drive T cells into tumors and increase their survival. So I'm thinking that in the future, we could do other immune cell screens, like for enhanced serial killing or enhanced engraftment efficiency. And we're pursuing single cell analyses for cells like these, which we think will help in the analysis. So really, you know, just to sum up what I've talked about, Loss of NF1 and SUS12 in human Schwann cells uncovers new therapeutic vulnerabilities. Better models are still needed and might be provided by 3D microtissues. And we've identified promising drug combinations. And then finally, the other side of the coin, using the immune system, we hope transposon mutagenesis might allow us to find candidate genes for enhanced adaptive T cell or other immunotherapies. So I'm going to stop there. These are the people that were involved in this work, and I tried to acknowledge them as we went along. So I'll stop there and take any questions you might have.
I've got microphones for any questions.
Great talk, David. So in the the middle of the talk, you talked about NF1 and SUS12, and you showed some really powerful synergistic interactions between drugs, which you came upon due to the NF1 and SUS12 aberrations, which sensitized to them. Then later you showed PDX models and patient models where those drugs were effective.
Yeah.
And I guess my question is, is that direct targeting, or are those on target effects? So do those PDXs in patients have the correct genomic aberrations?
They
or
do.
Are they off target effects due to a transcriptional state change, which allows these to be effective in the models?
So for all the PDXs, we know what genetic alterations are present, because the genomic RNA-seq and whole exome or whole genome sequencing are being done at WashU. Almost all of them are NF1 deficient and PRC2 deficient. A few retain the PRC2, but we haven't yet shown that the drug combination works in NF1 PRC2 deficient. It doesn't work in NF1 PRC2 proficient, which I think is what you're getting at. So we haven't demonstrated that for sure.
Are these tumors addicted to NF1 loss and PRC2 mutation?
Yeah, people have sort of done that experiment. If you add the gap-related domain back to an MPNS T cell line, or SUS12 back to an MPNS T cell line, it doesn't grow as well. It's less able to form a tumor. So they do rely on the continued loss of these tumor suppressors, which is different from other tumor suppressors, but they do, yeah.
When you were exploring the epigenetic alterations in your double knockout cells, you saw that global increase in H3K27 acetylation. Have you looked at the where yet?
Yes.
Where in the genome is chromatin opening and becoming activated?
Yeah, we have, and so we have ChIP-seq data on all these cells, these engineered cell lines also, and H3K27 acetylation, when you knock out PRC2, goes up kind of everywhere, but in certain genes it goes up dramatically, and you see the acquisition of new super enhancers. So it does seem to affect a subset of genes more than others. And so we have a list of genes that show dramatic increase in gene expression and dramatic increase in H3K27 acetylation. So we think those are the true targets then,
And then are any of those druggable targets?
Yes, some of them are. It's a variety of interesting genes, integrins, receptor tyrosine kinases like PDGF receptor alpha, MET, MER-TK. So there are some druggable targets that, based on that information. But again, it gets to this point of now we have too many drugs. So this is why I think, we developed, with Dave Wood, this idea of trying to do higher throughput studies with the 3D microtissues.
Fascinating work. I think one of the questions that I had as we were going through is the context dependency of changes. So for example, if you look at PRC2 in follicular lymphomas, it's almost exclusively associated, or loss of function of the complex is almost exclusively associated with EZH2.
Yeah.
And so I wonder if you have any thoughts on, as you're trying to hit these epigenetic modulators that are complex multi-protein complexes, where there seems to be some tissue specificity, what might you be thinking about in terms of potential side effects or, unanticipated things that happen when you mess up one component of the complex that might target one cell, but might have a completely different effect somewhere else?
Yeah. So I suppose there's the PRC2 is really complicated because this complex is promoting cancer in some cancers and inhibiting it in others. So it's actually more oncogenic in the B cell lineage. And that's why there's EZH2 inhibitors being developed for treatment, but it's a tumor suppressor in MPNST. So you can imagine that, in the wrong cell type, inhibiting EZH2, for example, is actually going to promote cancer. So, I think you bring up an important point. The effect of these epigenetic writers is context dependent. But the HDAC inhibitors have myelosuppression as a side effect. I don't know why, but I think in thinking about combinations that are realistic, we'll have to think also about non-overlapping toxicities. So.